Protein is a polymer of amino acid. Protein is a polymer of amino acid. Then how protein composed from amino acid? There are several types of protein like primary protein, secondary protein, and tertiary protein. In that primary protein or primary structure of protein, oxytocin is the example. In secondary structure of protein, we need to see that alpha helix, beta sheet, like beta plated sheet, beta parallel sheet. Also we need to see that 3 raised to 10 helix, collagen helix, pi helix, reverse turn, random coil. It is also important regarding competitive exam in life sciences. Also in tertiary, also we need to talk about secondary structures like motif, helix turn helix, and leucine zipper is also important. Then in tertiary structure, many protein have final structure is tertiary structure, that is composed of single subunit. Tertiary structure like myoglobin, cytochrome C, lysozyme, chymotrypsin, and ribonucleus. Then quaternary structure, many proteins that having more than one subunit, like we all know that antibodies are a protein composed from two different subunit. There are two light subunit and two heavy subunit is present in antibodies. Then also hemoglobin. In hemoglobin, two alpha subunit and two beta subunit is present. Then Rubisco is most widely present enzyme in Earth. Rubisco composed from 8 light subunit or 8 large subunit and 8 small subunit. In quaternary structure, we should taught different proteins like antibodies, Rubisco, then E. coli RNA polymerase, hemoglobin, etc. Protein stability forces including hydrogen bonding or various types of bonding in proteins. We also we also should know that Ramachandran plot. What is meant by Ramachandran plot, and in which how secondary structure involved. What is meant by allowed region and what is meant by disallowed region is also important in Ramachandran plot. Ramachandran plot is most important point regarding competitive exams. Then denaturation and renaturation of protein. How a protein can be denatured and how a protein can be renatured. It is also important. Then protein folding. In protein folding, chaperones and chaperonins are two protein are involved in protein folding. How chaperones can be fold a protein? There are two different types of protein also involved: globular protein and fibrous protein. Globular protein is soluble protein and fibrous protein is non-soluble protein. In protein folding, chaperones include HSP, heat shock protein, HSP 70, HSP 90, HSP 50. Then these all things should taught in when we deliver lectures. Then all things can be taught or should be taught. Concept of chaperones and its role in protein folding, also important. And also disease caused by misfolding. Disease caused by misfolding like Alzheimer's disease caused by misfolding of proteins. It is also important. It is all about unit first. Then unit second. Unit second, in which carbohydrate, lipid and vitamins is involved. Carbohydrate is most abundant biomolecules present on earth, and carbohydrate synthesized by plants through the process of photosynthesis, and that carbohydrate is consumed by heterotrophic animals or organisms. That's why carbohydrate synthesis is very important process, because mostly organisms are heterotrophic in nature, and all heterotrophic organisms is depends on autotrophic organisms for their food. Carbohydrate, its definition, carbohydrates function is also important. Carbohydrate is generally classified into three classes. One is monosaccharide, oligosaccharide, and polysaccharide. Monosaccharide is a simple sugar. Then in monosaccharide, we also need to taught what is meant by aldehyde monosaccharide or aldehyde group monosaccharide, and what is ketone saccharide, or aldehyde group presented when in which monosaccharide, it is also important. For example, aldehyde group monosaccharide like glyceraldehyde, erythrose, ribose, and also glucose, pseudoheptose are aldehyde group monosaccharide. And ketone group monosaccharide like dihydroxyacetone, erythrulose, ribulose, xylulose, fructose, and pseudoheptulose. These are all things can or should be taught when deliver a lecture. Then the basic term configuration and conformation is also important. The difference in between configuration and conformation. In second unit, it is also important that optical active carbohydrate, L form of carbohydrate, D form of carbohydrate, how we can say what is meant by L form of carbohydrate and what is meant by D form of carbohydrate. How a carbohydrate can be optically active, and what is the criteria to become a carbohydrate can be optically active. What is meant by chiral center? Chiral center is also important regarding optically active carbohydrate. Then also, here stereoisomer is also important. In first unit we also taught stereoisomer, and in second unit stereoisomer is also important. From that point some question will be asked. That's why what is meant by stereoisomer is also important, and how we can calculate a stereoisomer form or stereoisomer number in a particular carbohydrate is also important. There are a formula to count a stereoisomer number in a carbohydrate, that is stereoisomer is equal to 2n here, n for chiral center, n for chiral center. From that formula we can calculate number of stereoisomer in a particular carbohydrate. Dihydroxyacetone is a carbohydrate. It is not a optically active carbohydrate. Rest of all carbohydrates are optically active, but dihydroxyacetone is optically inactive carbohydrate. Polysaccharide, types of polysaccharide is also important. Homopolysaccharide, heteropolysaccharide. There are two types of homopolysaccharide is there. One is homopolysaccharide that is play a important role in storage of energy, and another is homopolysaccharide that is play a important role in structural composition. Here, some homopolysaccharides that play a energy storage role, that is starch, glycogen, invalin, is play a role in energy storage. And another some homopolysaccharide like cellulose, chitin, pectin play important role in structural composition. We should taught simple starch, then complex starch. Simple starch, that is amylose, and complex starch, amylopectin is also important. And how we classify simple starch from complex starch? Then glycoconjugates, proteoglycan, glycoprotein, glycolipids. Here glucose plus lipid, glucose plus protein, it is glycoconjugates. Lipids, definition, general properties and functions of lipids. Lipids is a heteropolysaccharide, lipids is a non-polar biomolecules. Lipids is a non-polar biomolecules, and in lipids, saturated and unsaturated fatty acid is present, or lipid can be classified into saturated and unsaturated fatty acid. And what is the criteria to classify fatty acids into saturated and unsaturated? It is also important. Classification of lipids, even and odd scheme of nomenclature, saturated, unsaturated, then polyunsaturated fatty acid. It is also important. Polyunsaturated fatty acid, that is PUFA, is also important because polyunsaturated fatty acid we take from outside. We taken out from outside. That's why it is essential fatty acid. Our body do not synthesize polyunsaturated fatty acid. That's why we can say that polyunsaturated fatty acid is essential. In that unit, we also need to taught what is meant by omega-3 family lipids, or omega-3 family polyunsaturated fatty acids, omega-6, omega-7 and omega-9. And from that unit, structure, general properties and functions of simple lipids, complex lipid, derived lipid is also important. In next point from second unit, that is vitamins. There are two types of vitamins. One is water soluble and another is fat soluble. Fat soluble vitamins like A, D, E and K, and water soluble vitamins like B complex and B enzymes and C complex. From that B complex water soluble enzymes, there are some enzymes from that B complex vitamins. There are some vitamins like B1, B2, B3, B5, B6, 7, 9 and 12. And that vitamins also have another name. For example, C vitamin is also called as ascorbic acid. B1 vitamin is also called as thiamine. B2 vitamin is also called as riboflavin. This name is also important because another name can be asked in various exams. Unit number 3, bioenergetics and photosynthesis. Bioenergetics, in which law of thermodynamics is also important. Law first thermodynamics law, second thermodynamics law. Then what is meant by redox reaction and its example. We all know that photosynthesis is the example of redox reaction. A redox reaction is the combination of reduction plus oxidation. What is meant by oxidation? Reduction, oxidative phosphorylation, oxidation plus phosphorylation. Architecture of mitochondria, mitochondrial structure and electron transport system or electron transport chain, its structure, electron transport chains, complex, four complex is present in electron transport chain, and how electron transport chain can be inhibited by various types of inhibitor like rotenone and rotenone, antimycin A can be inhibited to electron transport chain. Rotenone can be inhibited complex number 1 and antimycin A can be inhibited complex number 3. Uncoupler is also important. Then photosynthesis, general features of photosynthesis, evaluation of oxygenic photosynthesis. How oxygenic photosynthesis means what? Here only cyclic photosynthesis or cyclic chain is present. Here non-cyclic chain is absent. Then general photochemical events, light and dark reaction is also important in that unit. Light driven electron flow, photochemical reaction center in bacteria, pheophytin, photosystem 2, photosystem 1, photosystem 2, zigzag scheme, that is also Z scheme. Non-cyclic photophosphorylation is also important. ATP synthesis by photophosphorylation, how many ATP is invested and how many ATP is harvested is also important in that unit. Chemoosmotic theory is also important from that unit. Chemoosmotic theory or chemoosmotic model. Chemoosmotic model means when hydrogen transfer from stroma to lumen, then how many ATP is synthesized is also important. Unit number 4, bacterial chemolithotrophs, in which overview and modes of microbial metabolism in which microbes obtain energy and nutrients to live and reproduce. Microbes use many metabolic strategies to obtain foods. Carbon utilized bacteria, generally carbons utilized by bacteria in three different ways. For example, autotrophic, heterotrophic, and mesotrophic. In a heterotrophic way, carbon obtained from carbon dioxide. In heterotrophic, carbon obtained from organic compound. and in mesotrophic, carbon obtained by carbon dioxide as well organic compound. Chemoautotrophs can be divided into several groups like methanogens, halophiles, anamox bacteria, then thermoacidophiles, sulfur oxidizers, nitrifiers. These types or groups is also important in that unit. Physical group of chemolithotrophs, methanogens is also important in bacterial chemolithotrophy. And here is some references or some books for that paper, microbial biochemistry and physiology. Denninger, then Steyer, White and Smith, White and White is also important or very important for that paper. Cooper, the cell. That books is for particularly for third unit, that is bioaesthetics. Thank you, ma'am.
Thank you, sir. Hello, am I audible?
All DIMS and IPC. आठवायला 3 ला सर्व
Hello.
येतो ना.
हाँ. Good afternoon, all of you.
Good afternoon.
Respected Head of the Department, N.S. Joshi Madam, Chairperson Dr. S.S. Kanse Ma'am, Gorpade Sir, Uttekar Ma'am, once again, and all other faculty members. Once again, welcome to FDP program 2021. So I am going to express my views related to MSc 1 paper Essentials of Genetics. So MAMIT 104 Essentials of Genetics. काहीतरी होते. सगळंच गेलं. आता ते लर्निंग learning objectives. Students should be able to learn the essentials of Mendelian and non-Mendelian inheritance. Students should be able to study the basics of multiple alleles, essential genes, lethal genes. Students should be able to study properties, structure of the chromosome and their packaging. Hello, is it visible to you?
Yes, yes. It's visible over here.
So students should be able to study properties, structure of the chromosome and their packaging.
स्लाइड शो केलं तर स्लाइड शो होत नाही आणि ते परत माघारी येते.
So students should be able to know various repair mechanism in the body. Students should be able to learn about the pedigree analysis and various genetic disorders in humans. So unit 1, it is Mendelian and non-Mendelian genetics. So lectures allotted, there are 12. First is the Mendelism. So scientist Mendel, who is the father of the genetics, he has carried out some of the experiments on the garden pea plant, and while giving the result, he put forth some of the laws in the genetics, that are the Mendel's principle of the segregation, Mendel's laws of the independent assortment, and Mendel's laws of the dominance. And depending upon the character studied at a time, the two crosses are there, that is the monohybrid cross and dihybrid cross. So in this, or while explaining this monohybrid cross and dihybrid cross, the emphasis should be on the examples, and along with examples, students should be able to prepare the Punnett square, that is the crosses made between the plants or the different characters of the plant, and the result can be obtained. That is, the analytical view should be there, because this topic is very important for the competitive exams in all the life sciences exams. So for that, the along with examples, the genetic problems, or students should be able to solve that genetic problems. So next point is the epistasis, that is the when one gene masks the effect of another gene, it is called as epistasis, that is the genetic interactions. Statistical analysis of the genetic data, chi-square test is there. Then about the multiple alleles, ABO blood group system is there. Then essential genes and lethal genes. So in multiple alleles also, some of the genetic examples are there, and also in the examples of the essential genes and lethal genes, that is the genetic cross or example along with the genetic crosses are there. Then environment and gene expression, that is the codominance, incomplete dominance, and pleiotropy is there. So codominance, incomplete dominance. To explain this, some of the examples of the plants with different characters are there. For example, the plant with red colored flower crosses with the plant with the white colored flower, and which what is the result or outcome of that cross, that need to be explained. Then about the sex linkage, sex limited, and sex influence characters. So a slight difference between these three are there. So it can be explained with the help of examples. So this is about the unit 1 Mendelian, or first part of the unit 1, that is the Mendelism. Then another or next part is the non-Mendelian inheritance. The characters or which do not follow the Mendelian inheritance, they come under the non-Mendelian inheritance. So determining the non-Mendelian inheritance, then maternal effects are there. So whenever a slightly different ratio is there than that of the Mendelian inheritance, that comes under the non-Mendelian inheritance. Then maternal effects are there. After that, the cytoplasmic inheritance are there, that is the mitochondria, chloroplast, and infective particles. So cytoplasmic inheritance, in that the mitochondrial inheritance is there, or chloroplast inheritance, and infective particles means the kappa particles, which gives the killer property to the cell. So this is about the non-Mendelian inheritance. As it do not follow the Mendelian normal ratio, it is called as a non-Mendelian inheritance. So that can be explained with the help of examples. Then unit 2, that is the chromosome and their packaging. So structure of the chromosome, in that Lambert's chromosome is there, polytene chromosome. Then heterochromatin, that is defense against the mobile DNA elements. In that the heterochromatin, Lambert's and polytene chromosome, in that the structure is very important, and also the function is very important. Then heterochromatin, defense against the mobile DNA elements. So heterochromatin, that is the inactive form of the chromosome, or it is less active form of the chromosome. So it can be a defense against the mobile DNA elements. Means mobile DNA elements, they are the transposons or jumping genes. So whenever they uh, jumping gene they insert in within the chromosome, they bring about the inactivation of that gene. So whenever they insert inside this region, that is heterochromatin, they do not cause much difference. So in this way, there is the regulation of the gene expression in this heterochromatin. Then mitotic chromosome and their pattern, I think here should be meiotic chromosome. So slight change should be there. Mitotic chromosome and meiotic chromosome and their pattern. Then chromosomal DNA and packaging, in that prokaryotic and eukaryotic chromosome, unique and repetitive DNA sequences. Then about the nucleosome core particle, histone and non-histone. So here from the basic concept of the uh, nucleosome, that is the how nucleosome is there, and also the histone and non-histone proteins. Then about the ATP driven chromatin remodeling machines are there. So all that is important, or it is related to the regulation of the gene expression. Then about the covalent modification of the histone tail. So in that covalent modification, acetylation, methylation, ubiquitination, so all that different modifications are there, and also that modification lead to the regulation of gene expression. That is, some of the modification bring about activation of the gene expression, and some of the modification lead to the repression of the gene expression. So that must be uh, it should be understood by the student. Then split genes, exons and introns, that is the concept of the exons and introns. Then about the splicing mechanism, In that splicing mechanism, autocatalytic RNA is there and the spliceosome is there. So that is the mechanism of the splicing. It comes under this. Then the unit number 3, it is the DNA damage and repair. So first is the DNA repair, but I think before DNA repair, how the DNA is damaged, that should be understood by the student. Means before repairing the damage, how that DNA is damaged, and for that the mutagenic agents it need to be explained before repair mechanism. And for that the different mutagenic agents, actually it is already covered in BSc syllabus, but slight revision of mutagenic agent should be there. So after that mutagenic agent and the mechanism of the mutation, the next point is DNA repair. And in that DNA repair mechanism, error free mechanism is there and error prone mechanism is there. So error free mechanism, in that the mismatch repair, base excision repair is there, nucleotide excision repair, direct repair is there. So here some of the mechanism, they or a repair mechanism, in that the correction of that repair is there, or correction of the damage is there without breaking the bond between the damage or thymine dimer. And in some cases there is the breaking of the bond is there. So that two types need to be explained in error free mechanism, mismatch repair, base excision repair. In all that mechanism there is the slight pathway type, or some steps are need to be covered, and also some of the genetic diseases related to this mismatch repair, base excision repair, nucleotide excision repair, it should be understood by the student. Then error prone mechanism, in that error prone mechanism DNA recombination is there. So in DNA recombination, there are some models like holiday model is there, Mendelssohn's and Reddy's model is there. So that need to be explained. Then homologous genetic recombination is there. Then site specific recombination, that is the mechanism of the site specific recombination is very important here. Then eukaryotic transposons is point, uh, the point is in the syllabus, eukaryotic transposon. But before explaining this eukaryotic transposon, slight introduction of the basic transposon should be there. So what are the different types of transposons, and prokaryotic transposon, eukaryotic transposon, and mechanism of the transposition, it need to be explained here. So this is a unit number 3, DNA damage and repair. And after that, the unit number 4 is human genetics. In this human genetics, the first point is the pedigree analysis. So pedigree analysis, this point is also very important, or uh, genetic problems related to this pedigree analysis, they are very important. Or according to the different exams, some analytical questions are asked in this pedigree analysis. And here is the autosomal inheritance is there and X-linked inheritance is there. So autosomal inheritance, here the dominance, autosomal dominance is there, and autosomal recessive concept is there, and also X-linked recessive and X-linked dominant inheritance. So four main types of inheritance are explained in this pedigree analysis. So in that four main types, how the transmission of the diseases are there, and also the examples are very important, or which genetic diseases are transmitted by which inheritance, that is autosomal or X-linked inheritance. Then about the Lord score of the linkage testing. So Lord score means it is the ratio of the independent assortment and in linked genes. Then about the karyotype, that is the collection of the chromosome, and it is the ordering of the chromosome, arranging of the chromosome of an organism, that is called as a karyotype. So here some of the genetic disorders, that is hemophilia, color blindness, Huntington's disease are there. So genetic disorders, these three disorders, they are the example of this pedigree analysis, that is the inheritance, or from one inheritance type. The one example is given, that is the hemophilia, color blindness, Huntington's disease. So this is about the genetic disorder. So here, about the symptoms appear in this disease, and also the uh, transmission of the disease need to be explained. Then quantitative genetics, in that the polygenic inheritance is there, that is the uh, polygenic inheritance means it is the governed by many genes. Then about the heritability and its measurements, and QTL mapping is there. So QTL mapping is the quantitative trait loci. So this QTL mapping is done with the help of molecular markers like RFLP, RAPD, and some of the AFLP, some other markers. So with the help of that, the QTL mapping is carried out. So this part is covered in this human genetics. So in this part also, many analytical questions are asked. So that should be teach according to that aspect of the competitive exam, that is the genetic problems, or students should solve the genetic problem related to this all concept. So this is about the essentials of genetics. Then learning outcomes, students will be able to understand essentials of the bacterial and human genetics. Then student will be able to know structural levels of the chromosome. Then student will be able to learn about the DNA repair and genetic recombination mechanisms. Student will be able to learn pedigree analysis and various genetic disorders in humans. Then these are the reference books. Thank you.
Thank you, Pooja. Thank you so much.